So the bug is half the story, but the host is the other half of the story. And having seen the lineup for this course, which I thought was terrific, I thought I would spend some time on the host side, and was asked to say something about the kind of research that we do. And so it'll be a mix of some fairly basic stuff that you may already know, and then some stuff that's actually going on that I find very exciting and interesting, and a little broader than much of the life that I do, which is studying how the bug works. So if you'll bear with me, I have a lot of slides. Go fast, stop me at any point, and then we can have lots of time for discussion. So why TB? TB turns out to have been the largest cause of death in the history of the world, of recorded history, more than anything else that I know of. So if you look at the first paper ever written in English in epidemiology, it was 1632, one fellow named John Graunt, who did his epidemiology by running around to the churches in London and looking at the bills of mortality, what people died of in the various churches in a city called Westminster, which later became London. So 24% of deaths were early childhood deaths right after birth, and consumption was 1 in 5 deaths essentially. Curiously, grief was 1%, which tells you depression is not new, and murder was about the same as it is in Boston as far as I can tell. This is why people get TB. I'm often asked, what's the major risk factor for tuberculosis? The answer is breathing. When you look at this picture, you would think I'm showing it because all those little droplets are what you breathe in. Turns out they're too big. So it can only be about 5 microns to get into the lung. So multiply this by 100 to 1,000 times of tiny droplets. Each droplet can contain between 1 and 3 tubercle bacilli. So what we know is that a very small number of bacilli, maybe 100, maybe 400, is the dose required to cause a disease in humans. And what happens is it comes in through the airspace, gets picked up either by a macrophage, which as you know is a phagocytic cell, or mysteriously by this cell called the type 2 pneumocyte. And nobody knows really what it does and whether it is likely that it transfers the bug into other parts of the body. The characteristic of TB is these lesions, often picked up because they have calcium in them, as healed lesions or are translucent, which indicates there's stuff in there other than air, which would be black. So I like, this is, as you'll hear, I have a very good collaborator. We've worked together for 25 years, and I call this is tuberculosis in Los Angeles. We know it's TB because of the lesion. We know it's Los Angeles because of the bullet in the lung. TB is different than most diseases that are caused by infectious agent, not all, for a very peculiar reason. And you'll see in a moment. There is something called primary TB. You breathe in the bug, it goes into the lung, you get a disease, and at some point you either live and get a persisting infection or you die. That's the outcome of TB when it's in kids, which is relatively uncommon. There's a high probability of death. The case fatality rate of TB when it was untreated everywhere in the world was 50%. 50% of people who got TB up until the era of antibiotics died. The selective pressure on the human genome must have been enormous. So that's simple TB. Then some people don't die, but don't clear their bacilli from the lungs or in any form that you can detect. And they have chronic TB and are constantly spewing out TB and constantly coughing. There were something like 487 students and faculty at the Robin Hill School in St. Louis in the early 90s that got tuberculosis infection from a single teacher who believed he had a chronic cigarette cough and was actually a chronic spewer of tubercle bacilli. And then a more common form is an infection or recovery, and then no evidence of disease until some late time when you get reactivation. So again, in the era before antibiotics, there were two age distributions of TB. The peak in young people in this country is about 25. Somewhere between 19 and 30 or 32 would be when young people get TB and develop tuberculosis. Then there was a fallow period, and then TB rises in people who were in nursing homes. Presumably when their immunity starts to wear off, TB comes back, and there was a big peak of TB in the 60s. That's obviously changed with HIV, where the preponderance now in this country is either immigrants or people with immunocompromised immune systems. And then there are others that presumably have latent TB and they die and they never get clinical disease. How do we know they have latent TB? We don't. What we do know is they have a skin test, which is a measure of cell-mediated immunity that is caused by infection. So we know they were infected, and we can't predict at this point who's going to get disease and whether it will be acute or latent. Now, the curious thing about this disease, and one of the points of mentioning it, is some of the great work in the field was done before modern science really got involved. So as the major killer of mankind from infectious diseases, all kinds of smart scientists wanted to work on TB. And so some of the best work was done. You'll see this paper is 1948. You're going to see another one from 1931. And then when antibiotics came back and BCG vaccine came in, people thought TB was over. And most people in the US, when I say I work on TB, would tell me, oh, we still have a problem with TB? Yes, we have a problem with TB. So anyway, this is where TB attacks in a primary lesion. If you look at x-rays for where the bug hits and is walled off, it's all over the lung. If you ask the question, however, where is the active lesion when they get disease, it is almost always in the top of the lung. And there's no real definitive evidence as to why. The one thing we do know is that the apex of the lung has the highest level of oxygen. And TB is a bug that requires oxygen. So in any case, this is where lesions form. And when you dissect those lesions, what they look like is holes. And in the holes, there are actually three kinds of holes. I'll talk about two. One is a hole that is full of white garbage called caseous. It cuts in an autopsy like cheese. And that's a massive response trying to restrict the growth of the bug, wall it off, and vast numbers of host cells, generally macrophages and lymphocytes, get killed in the process and produce this absolute cheesy gunk. And that's a good outcome by and large. It just takes away a piece of the lung. The other is a cavity where there are bugs that live outside of cells. And when you cough, those are the things that are transmitted. And the curious thing about this disease is that it doesn't exist in nature. It's not sitting in the soil, it's not sitting anywhere. It is only transmitted from human to human, which makes it relatively unusual. And the reason you have to have lung lesions is if you didn't have lung lesions, the disease would have no other way to be transmitted. So the pathology is absolutely essential to the survival of the pathogen. Okay, so the history of TB is these are deaths per 100,000 in 3 major cities in the United States. It's interesting, CDC's scorekeeping on infectious diseases in the United States began in 1900. But in fact, there were these older studies in 3 cities that said it was about 400 per 100,000, which is a pretty high death rate from anything. So TB was a huge killer in the United States. And there's a curiosity, and that is this curve starts to inflect downwards in exactly the year that Robert Koch discovered the tubercle bacillus. And so I present this both because I think it's amusing and also to warn you about epidemiology, because the tendency would be to say, because someone found something in 1882, what you see following that must have been caused because of that discovery. Who knows? But it strikes me in a disease that has a long latent period, perhaps a lifetime, that this is an association that has to be purely coincidental, but it's an amusing coincidence. At any rate, from the time of Koch, who discovered really for the first time how not only TB was caused by a bacillus, but also how it was transmitted as an infectious agent. At the same time in Berlin, very famous pathologist named Virchow, who kind of ran the world, he was the super guru. He would not believe that TB could be caused by an infectious agent. He believed it was a cancer because of its big, chunky lesions in the lung. And when Robert Koch presented this on March 24th, 1882, he walked out of the lecture. He was wiped out. Okay, so TB was going down and going down and going down until the late 1980s. And I lived in New York in 1980. And there was a huge burst of TB. And this is a disease that really reflects a variety of things in the social environment. One is poverty. It is a disease of poverty. It lives where people are poor, and it's in every poor country. The second, it's a disease of crowding. The closer people live together, the more likely one individual will transmit to lots of others. And third, when you see a disease that's on the way down going up, there's something devastatingly wrong with the United States public health system to allow that to happen. And in New York, what we know is they cleared out all the psychiatric hospitals. 75% of patients in a great burst of money saving were put on the streets. And the Commissioner of Health received many, many prizes for his heroic effort in reducing the cost of mental health care. And these people simply became the homeless that really became very hard to find. And at one point they would go to a hospital, be given a prescription for which they had no money, and were expected to buy their own treatment. And what we know about TB is that this is one of the diseases, maybe the only one, where it is in everybody's interest for the state, the government, to pay for treatment to protect everybody else. So we had a huge increase in TB. And the good news is we've addressed that. A concomitant in the late 80s of this curve is HIV and the wearing down or the compromise of the immune system, which tells us that TB is very dependent on control by the immune system. Let me just say a couple things before we do something about the immune responses to TB. If you're infected with the tubercle bacillus, we can detect it to some extent, really poor test, but it's the only way we have, by a positive tuberculin skin test, which is just a ground up extract of the bacillus and the culture fluids. If you become tuberculin positive, you have a 10% lifetime risk of ever developing TB. Either highest risk would be in the first year or 5 years, and then the remaining 10% or 5% of the risk in old age when your immune system wears out and you hit a nursing home. If you have HIV, you have an annual risk of close to 10% of developing TB. So in developing countries, where almost everybody has been infected with the tubercle bacillus, and those that don't have active TB have latent infections, this is a sentinel disease for AIDS. And people who come with a cough are reluctant to go to a clinic because if they do, they're pretty sure they know they have TB, and they know if they have TB, they probably have HIV as well. So it is, from a public health management point of view, very tough disease to get people to want to show up for treatment. And the treatments we have, by and large, up until very recently, cure something like 95% of people when they're appropriately applied. We can talk about that in the discussion. So the focus of my work has been, for a long time, what are the necessary and sufficient immunologic mechanisms that are required to prevent or protect against disease? There are two kinds of immune responses in general. One only within the last 6 or 7 years was recognized, called innate immunity. And most of what we studied in immune responses has been something acquired, which is mediated by T cells, killer cells, regulatory cells, and molecules in the blood, immunoglobulins called antibodies. We want to dissect that out, figure what we need for protection. We know that the tubercle bacillus lives inside of macrophages, these scavenger cells that are designed actually to kill things. And yet this is the cell that this bacillus has chosen to live inside. So it has to have a lot of very smart defenses to protect itself against the killing mechanism of the most potent killer cell that we have for most infections. If it lives inside of cells that kill, and I told you that a cavity is when you get breakdown of the tissue, is there a mechanism that will kill bugs free? And the evidence is there isn't. So once you get through the host defense of being killed by the macrophages or having your lesion completely walled off, once you break that down, then you're in bad shape and you start transmitting and get sick. And of course, one wants to know how the bug causes tissue damage. And what one would most like to know is what could we measure that would predict whether someone's ever going to get TB or is resistant? Or the other way to put it, because the other thing I work on is vaccines. How would we measure whether a vaccine worked against TB if in fact in this latent period you have to wait 60 years or 70 years to know whether someone is protected or not? Very difficult challenge. Okay, just a quick slide to say that will have something to do with the end of the talk. Tough to study a lung disease in humans. You can't see what's going on in the lung. You can't go poking people and taking samples. So it's not altogether clear how the bug causes disease, and it's not altogether clear how we resist it. So there are a slew of animal models that people can put TB in, and it'll kill the animals. Mice, I told you it takes between 100 and 400 bacilli to infect a human. It takes 100,000 to 1 million to affect a mouse. So a mouse is not a terrific model for susceptibility because it is relatively resistant. Guinea pigs, one bacillus statistically will kill a guinea pig if it gets into the right place. Even more sensitive than humans. Very expensive to work with. Lousy pathology, so the pathology in neither of these really resembles in general human disease. Rabbits are terrific. They're exceedingly expensive, quite nasty to work with. They bite. And the relevant point is they have beautiful pathology, and they're really good to study something about pathology. Monkeys are quite susceptible, but there are two kinds of monkeys, macaques and cynomolgus monkeys, and they have very different resistances. So the bottom line of this is there's only one real thing, and it's tough to study, and that is human immune responses. And so from my business in this, from the early 80s up until about 5 years ago, almost nobody was studying TB in humans because it's so hard, and these animal models, as you will see, are so tractable. So we only knew when we began about the adaptive immune response, and an adaptive immune response is the main immune response everybody knows about. And that is that you have the capacity in your B cells and T cells, your thymic-derived lymphocytes and your bone marrow-derived lymphocytes, to have specific receptors that recognize specific antigens, and they can recognize virtually any chemical structure that's protein that you can put before it. How did they get made? A big debate for 100 years in immunology was, do the foreign proteins induce the specificities, or are they already there? And from some elegant genetic and molecular genetic experiments, the model that we all understand now is the receptors for everything exist on B cells or T cells before they ever see anything foreign. Just like when you go to buy gloves, you go to a store, there's every size of glove. All you need to do is put your hands in one till you find one that fits. That's actually how the immune system works. The receptors that are there, when a foreign protein, an allergen or an antigen, comes into the system, it finds the receptors it can bind to. And the impact then is to select those cells whose receptors are triggered to proliferate and make more of them. Bottom line is then, each immune response in the acquired immune system is a clonal response. One cell triggered by a specific antigen that binds to specific receptors, then clones that cell, and all the rest of the cells just sit there awaiting some other antigen to trigger that to expand. Bottom line, adaptive immune responses take time to develop. There are diseases like pneumococcal pneumonia, where if you don't have a pre-existing immune response, 7 to 10 days, which is what it takes to make an adaptive immune response, is about 5 days too late to survive. So adaptive responses are good if you have time to develop them. And we do that by riding a bus or a subway and getting what you would call subclinical infections, small enough doses of foreign bugs and antigens to develop low levels of immune responses that build up over time to the common pathogens. One of the worries in this world is we vaccinate kids, which is a substitute for childhood infection to protect against infection. But the question is, while we do that, kids don't get exposed to all these bugs anymore. And one wonders whether the increase in asthma, for example, is due to the fact that they don't get this kind of huge immune response when you get an overwhelming infection. On the other hand, kids don't die in this country from childhood infections, hardly at all. So we know that B cells have antibody receptors, immunoglobulin receptors, with specificity for a whole range of foreign antigens. And T cells have a different receptor that sees antigen, but not directly. B cells can just see proteins and peptides in their three-dimensional structure directly. T cells cannot see them. They can only see pieces of protein antigens presented by something called an antigen-presenting cell or dendritic cell, and I'll have a little more to say. These cells, both when they get triggered by an antigen, expand clonally and make a big response. It's huge, but it's delayed. And if you have an acute crisis of an infection, you don't have time unless you have pre-existing immunity. Okay, so a quick summary is the antibody response is made by B cells, and they get very, very big, and they become little factories for making proteins. They're called plasma cells. They make different kinds of antibodies. This is the antibody secreted into the gut, the vagina, the throat, the mouth. Helper cells are required to produce antibodies by B cells. So T cells don't produce antibodies, but they're essential. So it's a combination why. So one of the hypotheses is, why does it, the amazing thing about the immune system is it can see everything. Why doesn't it destroy the body? Why doesn't everybody get an autoimmune disease? And in fact, as you know, people do get autoimmune diseases. They get rheumatoid arthritis. They get glomerulonephritis. They get systemic lupus erythematosus. So the checks and balances aren't perfect. But the fact of the thing is, most people don't, and yet they can see everything foreign. And that is argued that you have helper cells and you have B cells because any one alone has the potential to go haywire and respond against host tissues, which Robert Koch once called horror autotoxicus. The fact is, it takes 2 cells to make a good antibody response. It's like having 2 keys to open a safe. You have to have specificity by this cell and by this cell if you really want to make an immune response. So in the cellular response, which is what kills tumor cells, rejects grafts, and is responsible for intracellular pathogens, it's largely done by helper cells, which produce molecules that work to activate macrophages and other cells, and they produce different kinds. This should be a gamma, okay? And there's 2 kinds of helper cells. Th1 produce interferon gamma and something called interleukin 2, and they activate macrophages and they help T cells that are triggered expand more and more. A second kind of helper cell produces another set of cytokines, and these are what helps B cells make antibodies. And then a third kind of T cell called a cytotoxic T lymphocyte is the cell that kill target cells. So with that, we're in a position to ask, how do you take a complex, multi-cell, highly diverse system, and how do you begin, in reductionist terms, to analyze what kind of an immune response is required to protect against TB? So when I began to do this stuff, there had just been a new technique worked out originally at MIT to knock out genes in mice. And we couldn't do science now without the ability to knock out genes. So what one did in the old days was made a piece of DNA that was homologous to the ends of a gene and put something that had a selectable marker, in this case neomycin, so that you would put it into 1 million cells. And if one cell had the gene in the right place and disrupted the gene so it couldn't make the protein, you could select for that cell by adding neomycin to the culture. That cell would survive, all the rest of the cells would be killed. And that's in fact how you make a knockout cell. You then do that in embryonic cells and you make a mouse. When you make a mouse, we were able then to say, okay, what are the critical molecules and cells required for protection in TB? The first of my questions, necessary and sufficient conditions. So these are control mice. They're wild type mice. They're perfectly healthy. And these are mice that had the gene for interferon gamma knocked out. 1 gene, that's all. And as you can see, these guys hang on for quite a long time after TB challenge, and these guys are dead in 3 weeks. Answer, interferon gamma is a necessary condition for protection against TB. Yes?
How is their survival without being inoculated in?
They would live perfectly fine.
No, the knockout. How does the knockout?
Perfectly fine.
Just like the wild type.
Just like the wild type. That's right. In a reasonably clean animal facility, okay? Mice very seldom show human pathology, in fact never, except when you have this kind of a knockout mouse. These are lesions in the liver, and these in the liver as well. And you see infiltrates of little dots. These are lymphocytes, T cells, and these big blobs are macrophages. And this stuff in the center is a necrotic lesion, the basic stuff that causes the cheese-like caseous necrosis in humans. So the model, for the first time, enabled us at least to see something that resembles human pathology. So this should say TNF-alpha. Going from Microsoft to Apple does not, it turns out, bring over symbols. We could ask the question, how do you get the pathology in the Ds? What makes the hole in the lung? What causes the necrosis? So everybody assumed that a newly discovered in the 80s cytokine called tumor necrosis factor, molecule that if you inject it into mouse tumors makes some of them go away, kills lots of things. Maybe that was involved. It induces necrosis in tumor. It mediates endotoxin shock and toxic shock syndrome. And we know it induces an enzyme called inducible nitric oxide synthase and production of nitric oxide and other reactive nitrogen intermediates in mice. So we thought that was fun to look at, and we expected that when we made the knockout of our mice, what we would have is long-lived mice because we had knocked out what should have been involved in the pathology. And lo and behold, the mice died. They died at the same speed as the interferon gamma knockouts, which was totally unanticipated. And the only inference you could draw is that TNF is necessary for protection as well as interferon gamma. And that was totally unexpected. And we did this either by an antibody to TNF in red or knocking out the receptor for tumor necrosis factor, so it couldn't work. Again, surprisingly enough, this is 2 weeks in a mouse lung after TB. The nice little holes there are nice air spaces, and these are early lesions with white cells infiltrating around a little bit of pink, which would be the bacilli. This is the control mouse. This is the one that we had anticipated wouldn't have pathology. This is the mouse that had a knockout in the gene for tumor necrosis factor, and it had a huge lesion, and all this pinky blobby stuff is again a caseous lesion. And what that said is that you can still make pathology by the bug itself or other cytokines, and the TNF was necessary in a mouse for protection, and not necessary for pathology, but in a whole mouse may well contribute to pathology in addition to the bug itself. So then the question we could ask is, okay, we knew now that TNF and interferon gamma made by T cells was absolutely essential for activating macrophages to protect mice, because if you knock them out, they died like flies. Everybody knew how polymorphs, white cells, kill acute bacteria like strep and staph. They produce a series of oxygen-free radicals. We also know that macrophages produce oxygen-free radicals when they're activated. So it was very clear what must be going on. We had only to prove it. So we tried to prove it, and we did the experiments that were appropriate. And the answer was the hypothesis again was wrong. So if in fact we took activated macrophages from mice, added took ordinary macrophages, activated them with TNF plus interferon gamma, we could get them good and activated. They would restrict the growth of TB. And then we could add inhibitors of hydroxyl radical, of superoxide, of hydrogen peroxide, and of singlet oxygen. And lo and behold, when we looked at killing of TB by measuring the ability of the bug to make RNA, none of them block killing. So here we had done the obvious experiment, taking the obvious mechanism and showing that whatever was killing TB in the macrophages, it wasn't the mechanism that everybody thought it was. What could it be? So we pursued that, and there weren't many other choices. The most obvious choice was the potential of these nitrogen radicals. And a third one that we paid no attention to was the guys working on fruit flies wanted to know why it is that fruit flies, when you grow them in these little bottles on agar, what protects them against the molds and bugs that come out of the stuff that we culture them on? And they found some genes in Drosophila that have antimicrobial peptides. But no one paid much attention because these are such powerful mechanisms, and we didn't know anything about it. So we decided to pursue the reactive nitrogen intermediates, and a good friend and colleague in Germany, Stefan Kaufmann, had worked out this scheme where an antigen or pathogen gets into a macrophage. It produces interleukin-12 that activates it, and that leads the T cell to produce interferon gamma that, in the presence of tumor necrosis factor, activates macrophages to produce, among other things, nitric oxide that can kill tumors or kill bacteria. So that's what we thought might be going on. And we did the experiment and proved that that was correct. So this is actually the first experiment anyone had ever been able to take a macrophage in a test tube and kill TB. They are about the toughest bug I know of for any immune or any other mechanism to kill. They're just hard to kill in a hospital, and they're hard to kill in a test tube. So what we did is took mouse macrophages and put them in a tissue culture dish, and we added TNF plus lipopolysaccharide, which is a stimulator of interferon gamma. And what we saw is a drop in about 50% in the viability of culturable colonies of these organisms. We know that the nitric oxide, if it's made, is made by this inducible nitric oxide synthase called INOS. And we know then how to knock it out with a compound called N-methylarginine, because the nitro comes from the epsilon amino group in arginine. Sure enough, if we added the inhibitor of nitric oxide synthase, the bugs grew. And if we added excess arginine that overcome the effect of the inhibitor, then the bugs are killed again. So this actually constitutes formal evidence that we had for the first time figured out how mouse macrophages kill TB. And we actually, in other experiments, figured out why oxygen radicals don't work, and that is because the bug has a huge set of carbohydrates and lipids on the wall that just soak up oxygen radicals. They're called scavenger molecules, and they simply inactivate the best that the cell has to offer. They also have enzymes like superoxide dismutase that chew them up as well. So that was very exciting and inelegant, because inhibitors seldom do what they appear to do, or selectively what they appear to do. So we knew the inhibitor knocked out INOS, but you could criticize it to say they might do something else. We don't know what else they could do. So a colleague in New York, Carl Nathan, and one of my former fellows, knocked out the gene for INOS and found that instead of living long and healthy lives out to here, the mice die just like other knockouts. But still, there is an interferon gamma dependent mechanism that if you knock out gamma, they still die quicker. So what it says is that if you knock out the ability to make nitric oxide, you have a really susceptible mouse. And this is confirmation that this is the mechanism by which mice macrophages kill TB. But even without that, there is still something dependent on interferon that in mice is still not understood. There must be a backup mechanism somewhere. Okay, now what we know is that that skin test that tells us that you have been infected with TB is really mediated by Th1 T cells. And it really is produced by production upon stimulation by antigen, by the acquired T cell response. It produces cytokines that activate macrophages, and that's why you get a big bump and creates a granuloma. But there is a problem, and the problem is that these are the protective efficacy of 18 trials of the BCG vaccine in different parts of the world. And it's the oldest vaccine still in use, and yet it has a problem. And the problem is that in some parts of the world, it has 80% protection, and in other parts of the world, it has no protection. And the only reason for showing you this, not only to say that BCG is highly problematic as a vaccine, is that everybody who gets BCG gets a positive skin test and a Th1 response. So this tells you that a Th1 response, interferon gamma and TNF, produced by Th1 responses, is a necessary but not sufficient condition for protection. That doesn't leave a whole hell of a lot of other mechanisms, but one that was obvious is killer cells. Is it possible that killer T cells might be involved as well. And there were some very interesting mouse experiments that were suggestive that killer cells might be involved. And this is an experiment in which we can get a knockout of a molecule called beta-2 microglobulin, which prevents killer cells from being recognized. And the experiment shows that if you knock out the ability to make killer cells, very interesting, BCG, which is an attenuated bovine tubercle bacillus, does not kill the mice. And that's pretty good evidence that it's really pretty attenuated. But the striking thing is these mice are still tremendously susceptible at an accelerated time to TB. But they don't die in 3 weeks. They die in 10 weeks. And what this tells us is that killer T cells are also necessary and not sufficient for protection. But they're not as major or as important as helper T cells in protection. So we have a complex business to say, in order to get sufficient protection in a mouse, it looks like you need helper cells, you need killer cells, you need nitrogen radicals, and you need the right kind of cytokines to induce them. And that seemed like we were getting somewhere. And the interesting part about the knockout of the killer cells is we saw another thing never seen in mice before. And I show it. This is a granuloma, a whole set of white cells loaded. You can't see it well. These should be red. All this dark stuff is tubercle bacilli. And what you see is breaking into a bronchiole, which is what would happen in humans. And that's how, this is what you cough up, is a lump of bugs, either singles or in a clump, that is breathed in and transmits. Mice never transmit TB to themselves because they don't cough, nor do rabbits. But in fact, we can mimic the human pathology by knocking out one part of the immune response. So about halfway through killing ourselves to understand acquired immunity, a colleague named Charlie J. Neway looked at a totally different phenomenon that it was very hard to believe existed. And what he was saying is, maybe forget T cells, forget B cells, forget clonal expansion. What do you do between the time you get infected and the 7 to 10 days or 3 weeks it takes to get an acquired immune response? Why don't you just keel over dead in those first 10 days from everything? And he hypothesized that there would be an innate immune response, something that was there in everybody that didn't have specific receptors for specific antigens, but had some kind of receptors that could see patterns of antigens, like repeating sugars in carbohydrates, like lipids that, at least to me, always look alike on a paper, with long carbon chains and whatever. And so wouldn't it be nice if in that 7 days you had, and all of us have, some basic native innate ability to recognize foreign things that have patterns, like bacterial cell walls, and that could activate through some signaling pathways, let's say macrophages, if those pattern receptors sat on macrophages. And if so, could they by themselves, without T cells perhaps, activate those cells to kill at least a small number of the offending initial bacteria in an infection? Can they kill intracellular bacteria? And can they begin to help expand an acquired immune response? So if they get out of control, in the innate response, you are helping amplification of the acquired immune responses. Part of this hypothesis says the pattern receptor response, the innate response, is there all the time and is not clonal, does not expand. It either kills what it sees or it gets overwhelmed. That's the basic hypothesis. Pretty hot area of science right now. And this is much of what we know about it. So it turns out on macrophages and many other cell types, there are toll-like receptors. TOL is a gene in fruit flies that controls resistance to funguses and to bacteria that contaminate Drosophila cultures. Drosophila people have been studying it for years. Nobody paid the slightest attention because who cares from fruit flies? What's it got to do with me? And it turns out it has a hell of a lot to do with me. So when you look at these TOL receptors, they're not specific. But for example, TOL 1 and 2 will see triacylated lipopeptides. TOL 2 and 6, which form a heterodimer, will see diacyl lipopeptides. TOL 3 will see double-stranded RNA, as in RNA viruses. This one will see lipids in most gram-negative bacteria. This will see tails of bacteria. This will see single-stranded RNA. And this will be called CPG repeats and islands. And God knows what these see, and there may be 5 more that I don't know about. At any rate, they then activate a common set of pathways that must have differences because you get different outcomes through a series of adapter molecules that then activate transcription factors that turns on some common and some separate sets of genes for all of these. And they're there all the time, and they cause everything from septic shock and autoimmunity to killing of bacteria inside of cells. So the question was, since this is such a tough bug, what do we know, what can we learn about innate immunity to TB in humans? What are the receptors? What molecule triggers it? How does the cell get activated? What are the mechanisms of killing? And what does it tell us about TB? Which is the rest of what I wanted to talk about. The bottom line is, with my good friend for 25 years and a whole slew of really helpful collaborators and a wonderful graduate student, we ended up writing a paper that pretty much brought the innate immune response to be taken as a serious component of triggering of the human antimicrobial response. And it is unclear, I will say at the beginning, that it has much to do with the mouse. And that's one of the points of the lecture. So we know that there's a lipopeptide on TB and many other bacteria called the 19 kilodalton protein, 19KD. If you look at bacterial counts and you simply take human macrophages and you trigger them in vitro for a day or two with this lipopeptide that we know, as you will see, activates the TOL2 receptor, this pattern receptor on mouse macrophages in human monocytes, then what you find is that if you activate the macrophages, they kill, this case, 2/3 virtually, of the input TB bacilli. And if you do this in a mouse that lacks the TOL receptor, you don't get killing. Formal evidence that this molecule works on a TOL receptor in a mouse, and if you knock out that receptor, you don't get killing. You can do the same experiment in human. We don't have a knockout human, but we can activate with 19KD, show we get killing by human macrophages from here to here. We can block it with an antibody to TOL2, and this is just a control antibody, so that we know that the innate immune system exists on both human and mouse macrophages, and when it's triggered by itself, by a TB product, it can activate the cell to kill. So the obvious question, we obviously knew how it worked in mice. We knew that nitric oxide was responsible for most of the killing in mice. If you knock out the INOS, then the mice die. So we did the experiment, and the experiment is very clear. If you stimulate mouse macrophages, you can produce nitrite, and you can kill the bacteria very nicely. You can also activate mouse macrophages, as you saw earlier, with interferon gamma plus TNF and produce nitric oxide and similarly kill the bacteria. So we got the mouse pretty well nailed. This is produced by T cells that activate macrophages to kill TB by producing nitric oxide. Now you do this in humans. And lo and behold, the general pattern of all of biological science, that what works in a mouse will work in a human, turns out not to work. So we stimulate humans with the Toll-2 ligand, nothing happens. We stimulate human macrophages with interferon gamma, none of it stimulates them to make nitric oxide. Not good. When we look at Toll triggering of human macrophages, again, we reduce the bacterial count. When we add interferon gamma, not only do we not reduce the count, we actually get a slight stimulation. So what works in mice here is not working in humans, and that's very puzzling and a big problem. So now if we activate the human macrophages, again with the Toll-2 ligand, we get killing in the mouse and we get killing in the humans. In the mouse, if we use an inhibitor of nitric oxide synthase, we block it, and in humans, it has no effect. And what this shows is the human response has nothing to do with nitric oxide. So we spent 10 years studying a mouse that has told us absolutely nothing useful about humans. That's the way science goes. So we had a phenomenon in humans. This is the first time anybody got human cells ever to kill TB in a test tube, and people have been trying to do that since 1890. So we've got a bit of a system to look at. And much of this work, most of it is done by my colleague Robert Maudlin and his students. I kibitz from the dean's office at the School of Public Health. At any rate, the bottom line is that you could ask the question, is there any reason to believe that the Toll-containing cells, we've just shown Toll activate cells in a test tube, are they activated in a real lung? And this is tuberculous lymphadenitis. These are actually lymph nodes in the lung from a patient with TB. And as you can see, the immunostaining shows that the cells infiltrating that have very high levels of expression of Toll. So we know that this innate immune receptor is really on the cells that come into lesions. So how do we analyze how that works? So there are two kinds of cells that we have to worry about, both derived in humans from a peripheral blood cell, the monocyte, which is a precursor that comes from the bone marrow, floats around the blood, and then when it gets into a tissue at the site of inflammation, either becomes a dendritic cell or a macrophage. And forget all the markers, but we can tell the difference. And the relevant point is the role of dendritic cells is to present antigen as well as cytokines, and these cytokines tend to help T cells in an acquired immune response to grow and amplify. But these are the cells that have all these little dendrites that are very good at fitting antigen into the receptors on T cells. You remember, T cells don't see antigen alone. It has to be presented by an antigen-presenting cell. The macrophages obviously can phagocytize very well, and the question was, could we show they had antimicrobial activity in a test tube? So these are the experiments. If you activate the macrophages, separate macrophages by their surface markers, macrophages that are stimulated by Toll have the ability to reduce the colony count of input bacteria, whereas cells not stimulated through the Toll receptor have the bacteria grow. When you do that with the dendritic cells that come from the same precursor, and you can never get them 100% clean, so this difference is almost certainly due to contaminating macrophages. These cells present antigen, but they don't kill. So now we have two cells derived from a common precursor in blood that now become very interesting. They got to be very similar, but one kills and the other one presents antigen. Can ask a really neat question, what's different? And so this is what's called the DNA microarray, and you can say, if you stimulate the cells with Toll ligand, both cells, you can get RNA and ask what genes are turned on, and you can then do an analysis of what genes are differentially turned on. Of all the genes turned on, which ones are turned on in dendritic cells but not macrophages, and which ones are turned on in macrophages and not dendritic cells. And maybe that'll give us a clue to how the macrophage kills and the dendritic cell doesn't. Okay, so each of these is a DNA pattern, And anything that's red says that the RNA for that gene was activated. And what you find is that there's a couple genes in here. Instead of 100, which is what happens in most DNA comparisons of similar cell types, there were only 3, 2 of which are the same, and I'll come back to that. Conversely, when you activate with the toll ligand, a different set of genes shows up in dendritic cells, but not in macrophages. So presumably, this few genes that is present here and not here has got to have something to do with killing. Forget this one. This probably does. We have a mouse knockout in that seems to be important in killing in mice. But this is a gene that knocked my socks off. So my friend Robert called on a Saturday morning and said, Bloom, we just did the experiment. There are only 3 genes, and 2 of them are the same gene. And I said, oh, that's great. What's the gene, and how does it work? And he said, the gene is the vitamin D receptor. What do you know about the vitamin D receptor? And I said, bloody hell, nothing. Haven't got a clue. No one has ever been able to link any vitamin to resistance to TB. What is this telling us? Well, my guess is this is telling us a lot. So the hypothesis was that toll will induce a pathway of activation of macrophages that requires activation of a vitamin D pathway. We know 2 parts of the pathway from other diseases. Vitamin D has to do with calcium metabolism, parathyroid function, kidney function, and it works as follows. That the hypothesis is that if you trigger toll, it takes vitamin D precursor in the blood, 25D3, which is not very active, puts a hydroxyl on to make the active form, which is 125D3. And this is what most effectively binds to the vitamin D receptor on lots of cells in humans. And what happens then when you activate the vitamin D receptor? Is it possible that it will activate a couple known products downstream that might have to do with resistance to TB? So the first of them, and most interesting, is a molecule that I had never ever heard of before that phone call, called CATHELICIDIN. It's been studied in Europe, called LL37, because it's a 37 amino acid peptide. And sequence is known. It's got plus charges in hydrophobic groups, probably dissolves in membrane and does something to membranes. And what the Europeans had shown is that if you put it in front of strep, staph, and E. coli in a test tube, it kills them. So one of the things that we learned is in the vitamin D pathway, there is a protein produced that has some capacity to kill bacteria. So human macrophages or monocytes were stimulated by toll and with increasing amounts of vitamin D3. And we showed that when you do that, you increase the messenger RNA and presumably the protein for cathelicidin. CYP24, this is the molecule that takes off the hydroxyl and makes it back to this. So at the same time that something goes up, you have a regulatory protein also regulated by vitamin D. And its job is to keep the vitamin D levels in balance. The second experiment was a mass spectrometry experiment done in California that shows that there is in fact production of the protein for cathelicidin and the right molecular weight of peptide when we had vitamin D3 in the medium. So we know that the killing molecule is produced by human macrophages in the presence of vitamin D and triggering of toll. So the question is, make the peptide and ask the question, will it kill this most difficult of all bacteria? And the answer is, if you measure either ability of the bugs to take up uracil into RNA, or you simply measure the ability to form colonies, then you get killing in a test tube. So we now have a human mechanism that will kill TB. And you can ask the question, well, we know that the tubercle bacillus goes into the macrophages in a very special vacuole. Is it killed in that vacuole? So this is fluorescent TB in a cell, and this is an antibody that stains the peptide for cathelicidin, and the answer is they co-localize in the same vacuole, and hence there is good reason to believe this molecule that kills in the test tube gets into the same place in the cell where TB really lives. Okay, and then by one of the most elegant techniques now available within the last year, you can block a specific RNA by the introduction of a small inhibitory RNA. And if you just measure either killing by viability uptake or CFUs, we can completely block the killing by blocking the messenger RNA for one protein, cathelicidin. So we think that is a key mechanism for killing. Now this is nice, it's kind of elegant science, and the question is, what's it got to do with real life, and why am I interested? What that phone call saying, what do I know about vitamin D receptor, meant is that when we got to this phase, there are some mysteries that have never been understood in the case of tuberculosis. And the first of it is, we know that African Americans are far more susceptible to TB than whites, and we have no idea why. We also know that when African Americans, before antibiotics, got TB, they had much more extensive disease, and we had no idea why. What we know is that African Americans have lower vitamin D, and the reason is vitamin D is made in the skin, and if you have melanin, melanin absorbs UVB light, and that reduces your ability to make vitamin D. Clearly, if you're in a sunshiny area in sub-Saharan Africa, wearing a loincloth all day, the impact of sunlight on melanoma and other bad things and overproduction of vitamin D led to humans developing melanin pigments that could filter at least huge doses. And blacks, for example, get very few melanomas. So we thought we had a really interesting and important finding that could be related to disease. We actually did the experiment. So we measured vitamin D in serum of whites, volunteers, and normal blacks, no TB, and they were at very much lower levels, 25% levels of whites, for levels of vitamin D3. And then we could ask the question, if we use the serum of blacks or the serum of whites to measure cathelicidin RNA stimulation, would they stimulate? And as you can see, there's a range, but on average, these were much less effective at stimulating cathelicidin RNA. So we know that there isn't enough vitamin D able to do that. We also know that if you simply stimulate with toll, there's not much production of cathelicidin if you don't have vitamin D present. So vitamin D is an absolutely necessary condition for the generation of an anti-tuberculocidal response. And a quick summary then of what we think is going on is toll stimulates the toll receptor. Toll receptor enables the active form of vitamin D to be made. It triggers the receptor, it makes cathelicidin, and presumably kills bugs. I would like to believe that, and I don't have any reason, and I wouldn't argue that cathelicidin is the only peptide or molecule that will do this in human cells. But I would say a couple things that are kind of interesting. So there's something called sanatoriums, and a guy named Bremer decided he was going to cure people like he was cured when they sent him off to visit the Himalayas. And he created the first sanatorium in Germany and allowed patients to be exposed to fresh air and sunlight. And people thought, as in this wonderful book by Thomas Mann called Magic Mountain, you spent 6 months or a year there and you either died or became cured. Again, Edward Livingston Trudeau also had TB and damn near died multiple times and finally did die of TB, as did his daughter. But he founded the first sanitarium, a lovely place in Saranac Lake, which is now a research lab. And he did a great experiment where he took rabbits at his lab at Columbia Medical School, and half of them were kept at Columbia Medical School, and half of them were put in the middle of Middle Saranac Lake, an absolutely gorgeous lake, where there's a small island which is now called Rabbit Island, and didn't infect them with anything. He just asked who survived better. And there was no question, the animals in the miserable conditions they must have been kept in the Columbia University Medical School Animal Institute died like flies. And these rabbits survived for years in their progeny on Rabbit Island in the middle of the lake, from which he inferred this was living proof that fresh air and sunshine was really a good thing and kept people alive. No one has paid any attention to any of that stuff at all. And yet, it is possible that being exposed less to fresh air, but to vitamin D and sunshine, may have had a beneficial effect, for which there's absolutely no scientific evidence because nobody asked the right question. So that explains one interesting thing. The second thing it explains is why a guy no one has ever heard of. I've only met one person who's ever heard of this guy, and he was president of the Nobel Committee in Stockholm. And his name is Niels Ryberg Finsen. He was a Dane, I think one of the only Danes who ever won a Nobel Prize. And he got the Nobel Prize for UV treatment of lupus vulgaris, which turns out, as you can see, to be tuberculosis of the skin. And so already in 1903, there should have been evidence that UV light could cure TB, but without a mechanism. And conceivably, it worked at some level by inducing local vitamin D synthesis. So what do we know about vitamin D? Well, we know that the levels of vitamin D are highest in a sunshine belt and decrease with increasing latitude. We're in one of the not so great areas to make vitamin D in Boston. If you look at pigmentation distribution in human skin color using a measurable scale, then you can see that most of the pigmented people live within these two tropic regions, which is the exposure to the highest level of sunshine. So clearly melanization is a response to the risks of high-level exposure to sunshine. Now if you look at vitamin D status, that what you find is old world primates have high levels of vitamin D for whatever reason. In this phase of evolution, we didn't. But if you take 4,000 units a day of vitamin D, even though we're here, we can get up to here. But most of us don't. And your one-a-day vitamin has 400 units, 1/10 the amount to make the slightest difference in blood levels. Oh, God. Right. Okay. Now, one of the curiosities, small aside, is how is pigmentation determined? And one of the astonishing papers is a paper in Science. There are more genes now, but this was the first of them to be discovered, a gene I have no idea what it does. It's called SLC24A5. And it's a gene that was knocked out in something called golden zebrafish. And the zebrafish lose their stripes. And the question was, how does that work? And is it any relevance to humans? Well, who would have even thought that a zebrafish gene would have relevance to humans? But in fact, it does. And this is the major gene in humans that determines the degree of melanization of skin. There are other genes that do, but this is the main one. And it doesn't do it by blocking synthesis of melanin. What it controls is the size of the melanosome. the granules that contain the melanin. And so if you have variants or polymorphisms of this gene, and they're in sub-Saharan Africa, you have big melanosomes, and if you're in the northern parts of the world, you have tiny melanosomes in smaller numbers. So it's kind of interesting that this is a very important evolutionary distribution, and it is likely that the regulation of this evolved a long time in humans. Now this, as you will see in the next slide, is the skull bone of someone who has pits in the skull that is very likely to be caused by Pott's disease, which is TB of the bone. And this is an archaeological finding, as you will see momentarily, a zillion years earlier, and must be very early. A 500,000 year skull is the earliest sign of TB that I'm aware of. And presumably, dark skin of our ancestors protected people from UV radiation, and this poor fellow had TB. How important is vitamin D to evolution of humans? Well, one of it is that it obviously, if we're right, has something to do with protection. But it doesn't matter whether we're right or not, because if you don't have enough vitamin D, you get rickets. And one of the curiosities of this pelvis is no child could be born through that pelvis. So you have a wonderful Darwinian selection against women in pregnancy that have low vitamin D, have a low probability, or a lower probability, of having deliverable kids if they have rickets. So if you grow up in sub-Saharan Africa and you get enough sunshine, you're not going to see this. If you're in the north and don't, it could be very serious. So this adds to the selection value of making vitamin D, in addition to its resistance to infection. Finally, when Everett Livingston Trudeau brought TB sanitarium to the United States, the question is why they didn't work. Here there were these people, they were sent up to Saranac Lake, New York, and all over Massachusetts, and famous people used to go. So the reason they didn't work is they put windows on them and glass filters out UV light. So they never got enough UV light to make enough vitamin D. Before I get to how general this finding is likely to be, I haven't mentioned why we have results different than mice. Maybe I'll save that for the last slide. At any rate, so the question is, this is great for TB, maybe it's involved, maybe it's not. Just recently, the CDC published the data on staph infections by race, and the data are absolutely striking, white and black at various ages in JAMA, or in the CDC report by age. Blacks have a much higher susceptibility to TB than whites. No explanation. Is it possible that this is a general microbicidal mechanism that works not just against TB, but many other infections? And finally, these are data from Ed Giovannucci at the School of Public Health here, who's been studying colorectal cancer and prostate cancer. And these data would suggest that if you simply measure the correlation between blood levels in people in the Boston area and around the country, blood levels of vitamin D, then if you have a reduction of, if you have 34 nanograms per mil, which is low, you have a 50% increase in risk for colorectal cancer. That is, there is a very striking relationship between the amount of vitamin D in serum and the odds ratios, which is shown here, the chances that you will get colon cancer. Unclear what the mechanism is by which vitamin D, which associates with colon cancer, with prostate cancer, and with breast cancer, what the mechanism is. There has never been a mechanism. One wonders whether the cathelicidin that produces this peptide inside of cells can leak out and kill tumor cells, and no one's ever looked. Finally, it explains why people made a good living in the 1920s selling bottled sunlight, which turns out to be cod liver oil, very rich in vitamin D, which I must say I was forced to take as a child, and it's horrible. So we have a model where we would argue that in the innate immune response, we can get human macrophages to kill TB, which is very much harder, at least in a test tube, to do with the same mechanisms that work in mice, and it works in the absence of detecting oxygen radicals or nitrogen radicals. One of the few differences that I'm aware of between human and murine antimicrobial responses, and if you think about it, it turns out mice are nocturnal animals. They don't like the sunshine, they don't go out into the sunshine, and they don't make a lot of vitamin D. Cows don't. Fetal calf serum has no vitamin D, so most people would have missed this if they did studies with fetal calf serum and not human serum, which has vitamin D. So that's important, and the bottom line is, if you're going to study, the proper study of mankind is man, there are times when you really have to work on the real case. Models are good only to a point. Since vitamin D controls calcium, what you see in an X-ray is calcium. It's got to be involved in the lesions. It may explain why sanitaria once worked till they put windows on them. It explains the 1903 Nobel Prize that no one's heard of in medicine. It explains why bottled sunlight sold very well and probably would sell once again if it was allowed. You can't even buy in a pharmacy 4,000 units or 2,000 units a day's worth of vitamin D. You have to get it from a nutrichemical place or a health food store if you can find it. And finally, the real possibility is this doesn't control just TB, but it may actually explain why blacks and some Asians have higher risks for TB and possibly methicillin-resistant strep or staph infections and colon and other forms of cancer. What these experiments beg is there are no clinical data to know whether this would actually work in people at high risk like blacks. So we would hope to have funding from somebody to do a clinical trial to see whether contacts of TB patients in Tanzania, for example, that we know have low levels of vitamin D, given for pennies a day, vitamin D may be able to develop resistance and protect them against TB. So with that, I thank you for your attention, and I apologize for going over. My last line, this is one total waste of time. So Robert Koch discovered the tubercle bacillus on March 24th, 1882. Virchow walked out. He was the biggest hero of the time. He was written up in the London Times within 48 hours, and the Herald Tribune in New York had a front page story on the discovery of the tubercle bacillus in 1882, and he was the big cheese. Meanwhile, there was a guy no one's ever heard of who was the great tesiologist in Italy. A tesiologist is a TB expert. That's all he saw was TB. And he has this wonderful line that I like a lot, which is the bacillus is not yet all there is to tuberculosis. In historical judgment, I'm not sure that Bacelli wasn't righter than Robert Koch, but at any rate, that's for debate. Okay, time for questions, please.
Yes, is there any indication that variations in vitamin D synthesis are related to different numbers of dendritic cells versus macrophages coming from monocytes?
It has, to my knowledge, no effect on the development of macrophages from monocytes. There are other lymphokines or cytokines that are necessary to do that. One of them is GM-CSF, for example. But no, it only works on the macrophage or the dendritic cell that already exists. Yeah,
Okay, now you totally scared me to death.
I hope so.
You have, because my vitamin D level is non-existent. And they've been treating me with mega doses of vitamin D.
How much doses? I bet.
Say
I thought-
10,000
That's a lot.
units once a week, along
That's
with
fine.
vitamin
Good
D
for them.
pills every day.
Great.
But they never mentioned any of this. They're talking about bone thinning.
Because vitamin D is terribly important for calcium deposition.
Right.
And getting the dose right, I mean, too much can give you bone where you don't want it. But certainly most women in this country are hypovitaminemic, or at least in the northern latitudes.
So I'm more susceptible now to TB.
No, not if you're taking your pills.
Okay,
They're doing good.
but what they
They
want
don't know
is-
why they're doing you good, but they're
Right.
doing you good.
But they want to take me off that, and they don't know why my vitamin D level is so low.
How much time do you spend in the sunshine? Not a lot.
I mean, average. I don't know why this has just happened. I mean, I never seem to have had this problem before.
no idea. Well, nobody ever looks for vitamin D,
Yeah.
right? Somebody has to look.
So what would be, why don't they take blacks, for example, and just give them these supplements?
Because no one has given us approval yet to do the clinical trial. This is all within the last year,
Oh, okay.
okay? So, and you know,
Should have like a dairy company fund it.
You know, you can get it, but only from health food providers. So I take 4,000 units every other day. You're taking, my friend Robert Maudlin takes 10,000 once a week. You can do it. It's very cheap. We have no DNA similarities, so the gene structures are different, and the amino acid sequences are all different. So it's very tough to find them by using homologies either at the gene level or the protein level. What they have in common is they're both cationic. They bind to negative surfaces. Most bacterial cell walls are negatively charged, so that would make a lot of sense. And most of them are these so-called amphipathic alpha helices, which means when part of them is lipid, part of them is hydrophilic. And that means when they hit a membrane, many of them fold into helical structures that insert in the membranes. Some of them form hairpin structures that insert in the membranes. And the likelihood is they function by producing pores in the membranes. And I also could have shown a slide. When you add it to the tuberculosis, which I told you has this really tough, waxy cell wall, it develops blebs and bubbles and pops. So they almost certainly work by making pores in the membrane and allowing the media, which would be serum in the body or tissue culture medium, to invade the cell. Cell can't control its permeability and just blow the cell up by osmotic pressure. So they're almost all membrane active. Yeah.
This is kind of interesting to me because is there any relationship between susceptibility of people who have tuberculosis getting melanoma or vice versa? Because my family has a history, one of the highest histories in Massachusetts of melanoma. My father had 6 brothers and sisters, all of which have died of melanoma, and 2 of which had tuberculosis. I've tested positive for melanoma genes. So I'm just curious.
It's a great question. No one has looked. And the prediction would be if you have low vitamin D and if this mechanism had anything to do with melanomas. I told you Ed Juvenich is an epidemiologist. He's only looked at colon, breast, and prostate. I would doubt that he has any data on melanomas, and it would be a great thing to look at. And if 2 of them had TB in the family, it would suggest that it may be involved in protection against melanomas. And it's interesting, blacks don't get melanomas. Very interesting.
What's the downside of taking 4,000 units a day?
It can kill your kidney, it can kill your heart, and it can put bone in places you don't want it. So, so you don't want to take infinitely high amounts. But what we know is that the stuff in a 1-a-day vitamin isn't going to get a detectable increase in blood levels. 1,000 a day isn't really going to do it. So we have a colleague who works in North Carolina who wrote a review in the New England Journal within the last year on vitamin D. and his name is Hollis. And he feels the ideal dose for Americans that don't see a lot of sunshine would be on the order of 2,000 units a day, 4,000 every 2 days, or 10,000 a week. And you can buy them at food stores. The only caution there is you ought to check somewhere to see if what they're selling is really what they say it is, because there's no FDA control on health food stuff.
Could you get a small pill? Take a lot of them, right? Could you get like a small dosage of vitamin D and just take a bunch of it?
You could. It's just easier to take the right amount, you know. Yeah.
2 questions. First, do you know how much time you need in the sun to produce your daily vitamin D?
I should, and I don't. It's relatively short, depending on the intensity of the sunshine, a couple hours.
A couple of hours?
Yeah.
Second question is, about 4 slides back, you had that showing that huge increase in the black population. I think it was MRSA you were looking at. And I'm just wondering if, it looked to me, right, what hit you was, that's a time when people are going to work, you finally become an adult. And when they were young, it wasn't so bad, and all of a sudden it increased. I'm just wondering, going to work means inside. Were they even decreasing their levels of sunlight, vitamin, and is that just the relationship?
Could well be. No one who wrote that chart has related that to vitamin D. I mean, they haven't either read our paper or it hasn't sunk in yet. You have to have 100 papers in anything before anybody will actually know you've done anything. Yeah.
I have 2 questions. I was just wondering if sunscreen is an issue?
Yeah, it cuts down the UVB, and it's a good thing to do at some levels. For melanoma and skin cancer, tremendous impact of sunscreen on reduction in melanomas in Australia. So the stuff really works. It depends on how much exposure to sun as to how much vitamin D you'll get. And you need a lot less if you're being exposed to the sun and are not melanized to get in a proper level of vitamin D than sitting out all day on the beach. So it's, no one has looked to see if you put on, sunscreen at 30, how long you have to sit in the sun to get your vitamin D's level. There are no data that I'm aware of.
My second question, I was recently speaking with a friend who does research on multiple sclerosis, and he was saying that one of their projects right now is looking at vitamin D levels and incidence of multiple sclerosis. I was wondering if you've heard of that.
I'd love to know what the hypothesis is. It would not cross my mind why that would be obvious.
The latitude.
Oh, the latitude. Yes, absolutely right. Yeah, you're absolutely right. Yeah, absolutely right. So MS is really low in tropical areas in the vitamin D belt, and it really increases. You're absolutely right, for which there's never been any molecular explanation.
They're just starting to do
That's a great study. You're absolutely right. Yeah.
So, in your estimation, would vitamin D, the administration of vitamin D, be more involved in preventing tuberculosis?
Great question.
Or in actually reversing it?
Yeah. These are all terrific questions, I must say. I'm really excited by the questions. So the question is, what would be the role of vitamin D? And there are 2 ways of looking at it. One is still hypothetical, but it makes a lot of sense to me. And the question would be, what would you expect vitamin D to do for you? Or more generally, what would you expect innate immunity to do for you? And so what I said in the beginning, and why I said it twice, was the multiplicity of infection. The number of bacilli that it takes to give someone TB is on the order of 100 to 400. It is very easy for me to imagine that some of those get through to those pictures in the lower lung. And you have enough macrophages, if each macrophage eats 1 or 2 bacilli, okay, and they trigger the toll receptor with their own lipopeptide, that they could wipe out very low numbers of bacilli, okay? And that's a kind of first line of immunity to a low infection number. Once you have a bigger number or get past what the macrophages can do, you're in deep trouble, unless you create an acquired immune response and you develop lots of T cells and they have a mechanism to activate the cathelicidin pathway. which is what now Robert's lab is working on. And the evidence is they do. And what we know only is you don't need toll, but you do need a T helper produced cytokine called you need a macrophage produced cytokine called interleukin 15. Macrophages produce interleukin 15 when they're activated by interferon gamma itself. So you'd have to have an acquired immune response. T cells would activate macrophages that are not themselves able to kill or produce cathelicidin. That then would produce IL-15, which would then enable other macrophages in the vicinity to activate the cathelicidin production pathway, and that much they have shown. So what you have is a small number of T cells and a small number of macrophages that get properly activated. They then produce IL-15 that can activate all the macrophages in the neighborhood. So what we're trying to argue, and they will take lots more data, is that my guess is the best you can get out of innate immunity is in the short term when you get an acute infection, you either live or die. You either kill them all off or they get past what the pre-existent innate mechanisms are, in which case you need an acquired immune response. And it may slow things down a bit to enable you over 7 to 10 days to develop that response. For us now, the key question is, when you do have a really acquired T cell response, is it the same mechanism, or do you make nitrogen radicals, oxygen radicals, and whatever in humans? No one ever thought the human would be different in the mouse, so we really don't know. But the guess is that that's the way you would handle very large numbers of bacilli. So in my own view, how would you do the clinical trial? Let me ask you. So if we wanted to give vitamin D to people in Tanzania, who would you give it to? Would you give it to people who already have active TB? Would you give it to their contacts, most of whom have probably already been infected, but are either latent or resistant? Would you give it to babies the day they're born because they have no bias and you could soup up their innate immunity? Who would you give the drug to?
They're all great.
They're all great, and I think the least likely is the one we're likely to be able to be allowed to start on, which is people who already have TB. And the problem there is you have to treat them. And so you have 3 really strong or 4 really strong anti-TB drugs, antibiotics. Are we ever going to see an effect of adding a little dinky once-in-a-day vitamin? I'm dubious we'd actually see an effect. A little more optimistic in their household contacts, where they're healthy. Right. We want to keep them healthy versus a control group that we're not going to keep healthy or we're not going to give vitamin D to. Yeah.
Do the full spectrum lamps that were popular a couple of years ago, do those stimulate vitamin D production?
I have the vaguest idea. All you need is UVB, and I don't know the wavelengths.
Could you speak to how treatment does work in the sense that, is there a correlation with how those drugs work against TB? And I guess multi-strain resistant TB too is kind of another whole area of like what's the difference between those as well. They're just going after the bacteria and not really sort of going to wipe it all out in the body.
So it's a really great question. That's another whole lecture, but I'll try to summarize quickly. There's the drugs used to treat TB actually kill TB that divides. Okay, and that's how you screen for drugs. You put them in a machine called the BACTEC machine. It gives them good culture conditions. You test and screen antibiotics, and you ask which ones will block growth of TB. And in people with active disease, that's what's going to kill them, is the overgrowing of the bugs. So there's not a shred of evidence that any of the antimicrobial agents has any effect that we know of on the immune response. The drugs are good. They should clear up, even in AIDS patients, simple TB to the tune of about 85 to 90% in AIDS patients and 90 to 95% in non-AIDS individuals that have TB. They work very well. And they work at various levels. They work on RNA polymerase, they work on transporters, they work on other molecules. Just a small, okay, so that's the first piece. One of the incentives to try to understand immune mechanisms is we're running out of TB drugs, okay, just as we ran out of malaria drugs. And that is when they're misused, you get resistance. So the history of this is in 1952, Selman Waxman became very famous, won a Nobel Prize, because he discovered streptomycin, a fungal product, one of the first that was screened against TB and killed TB and was used as a drug. So he was hailed all over the world as the guy who really discovered, again, front page New York Times and every other newspaper, the cure for the major infectious cause of death in the world. That lasted for 4 years, and then Selman Waxman is the author on a paper that shows patients treated with TB became resistant to streptomycin, and the bug just grew happily. And in fact, some of them had bugs that are streptomycin dependent. If you don't put streptomycin in the medium, they don't grow. It's turned out to be an interesting tool for animal studies. So we know that if you apply to any kind of significant pathogen a single drug, unless it kills it very fast, you run the risk of resistance. And the number of, again, there's a famous set of experiments that ask the question, does the drug make the bug resistant by exposure, or are bugs already resistant before you add the drugs? So what's the answer? Prior resistance, exactly right. So there's 1 in 10 to the 7th resistant to almost everything. And what you do, again, is select for those that are resistant and kill off everything that's susceptible, and that's why you amplify it. So we know monotherapy in TB in 4 years didn't work. I would point out that up until last year, people were still treating malaria with one drug. And it's not a great surprise. We've run out of all the drugs in malaria. So there's one left called artemisinin, and WHO has now made it as a policy that you should not give anything but combination therapy for malaria. And this is not widely being done because people are selling one drug cheaper than 3 in poor countries. So it was a principle that was picked up in '52, and for most other infectious diseases has rarely been generalized because it adds to cost. Okay, so now we have a phenomenon of multi-drug resistance. So there are 3 drugs that are really used to treat TB. One called isoniazid, one called rifampicin, and one called pyrazinamide. The first 2 are microbicidal, and they're very good. In Russia, somewhere between 70 and 80% of the patients are already resistant to isoniazid. That means when you treat them with 3 drugs, you're really only treating them with 2 drugs. And one of them isn't very good, pyrazinamide. So you're really treating them with 1 drug, which means you're going to end up making them rifampicin resistant. And now you're creating, by treatment, multi-drug resistant TB. And since they're the 2 best drugs, once you have that, it's very tough. We have an armamentarium of 9 lousy drugs, utterly toxic, can get people through. Paul Farmer and Jim Kim at our school in the medical school fought with WHO, who chose not to treat AIDS patient with MDR-TB because they just thought it was too expensive and they're going to die anyhow. And they proved they don't have to die. They have to get a whole mix of secondary, 2nd and 3rd line drugs. So it is inevitable that, I should say one other thing, the way you treat TB is not like 10 days and come back and I'll pronounce you cured. It's a minimum of 6 months treatment. Compliance. When people are sick with TB, they have pains, they cough, they're sick as hell, they have fevers and chills. They'll take anything for a month. And 70% of people or more feel great at the end of a month. And that's a bad thing, because then they stop taking their pills. And then they come back 2 months later with a relapse, and they take them again, and the next relapse, they have drug resistance. Okay? So compliance is really, really difficult for any treatment. Heart disease in the US or anywhere, and getting people to take 3 drugs or 4 drugs for 6 months, or if you're an AIDS patient, 9 to 12 months. And every time a patient stops taking drugs or is not given appropriate therapy, you run the risk of creating resistance. And if they have resistance to one drug, you simply select for resistance to the lot. So things were bad, but we could get around those relatively uncommon cases of MDR-TB. Then in KwaZulu-Natal last year, at a place called Tugela Falls, a whole slew of AIDS patients died within 25 days, which no one has ever seen in tuberculosis, of TB that was unable to be killed by anything. And no one has seen people die that quick. And that turns out to be what's now called XDR-TB, extremely drug-resistant TB, which is resistant to almost everything. And if that gets out of the bottle and gets from AIDS patients to non-AIDS patients, just think about the medical personnel that are exposed to these people, the nurses and the physicians. We don't have drugs for them. It's scary. So the good news, in a way, is horrible. Epidemiologically, they died in 25 days. They didn't have a lot of time to spread the infection. But it's a very serious business. So this is, to go a long-winded answer to your question, this is why people have to, I think, take the immune mechanism seriously. Because it doesn't matter whether we're killing MDR-TB, XDR-TB, or plain TB. The immune system doesn't discriminate, you know, the mutations in the bugs that have to do with their ability to grow with antibiotics. And we're going to need a better vaccine than we have, and that's a problem. But the vaccine we have is not terrific.
I was just about to ask about a vaccine and wondering why has it been so difficult to get a good vaccine?
Sure. I think it's the first question I ask. What are the necessary and sufficient mechanisms of protection? And the answer is we don't know. We know that T cells are necessary. We know that helpers and killer cells are necessary. We have a pretty good idea that antibody is not necessary. But every mouse that's immunized with the best we can make for vaccines will sooner or later die of TB. So whether a mouse is a good or a bad model, and I'm arguing it's not a great model for humans, they all die. 90% of humans infected with TB who become skin test positive don't ever get clinical TB. So the challenge of a vaccine is not so bad. It's to take that 10% whom we can't identify and push them over the threshold and make it 100%. And the only way to do that is to understand what the mechanisms of resistance that are sufficient are. For example, is cathelicidin really important for resistance? I don't know. We've done all the work we've done. It's a new mechanism. We'd love to run the vitamin D clinical trial. If it worked well, we'd have some reason to believe it might work. We don't have, so I've shown you the 18 clinical trials. In the UK, BCG in adolescents was very large numbers, found to be 77% protective. One of the two same vaccines was used in India. It was 0% protective at every age. So we don't have an explanation for these geographic differences. We don't have a proved hypothesis. We have a lot of hypotheses, and I could discuss those. But the bottom line is BCG works very well in some parts of the world and very poorly in protecting adults from TB in other parts of the world. But in all parts of the world, it protects kids, or it seems to protect kids from disseminated TB that kills them. So it's not worthless, and that's why it's given at or around the time of birth, because it keeps kids from dying of TB. We need a better vaccine, but the problem is, unless we have a surrogate measure of protection, who's going to wait 60 years to see if your new vaccine works? And that's what it takes with latent TB to know whether someone's going to die or not. So we need to know whether it's cathelicidin or nitric oxide or some other endpoint that our vaccine did what it takes, just like developing an antibody that neutralizes polio predicts that if you have antibody that neutralizes polio, you're not going to get polio.
The spread of TB, when you had the first picture of breathing, so for TB to get into your lungs, you're breathing it in just through the mouth.
Now, you also showed pictures of bone and cutaneous TB though, too.
Probably not transmitting. So the other interesting thing, a lot of AIDS patients get extrapulmonary TB, and that's a problem at a couple levels. First of all, they're going to die. TB of the kidney, TB of the spine, TB of the brain. But because they're immunocompromised, you can't often see a positive skin test, so you don't know they've been infected. All you do know is if you get them to cough, you won't find any bacilli, so you can't make the diagnosis, because the diagnosis is made by discovering bacilli in the sputum. So we don't have we have no good diagnostic test for anything but people who already have lung infections. We have no diagnostic test for people who have extrapulmonary TB, and we have no diagnostic test for anybody who has latent infection.
It's not transmitted, and you said that it's not found in the environment. Where is it coming?
It's transmitted by breathing.
But, I mean, for the cutaneous or the...
Oh, they don't transmit it, presumably.
They got it.
They got it.
So it initially was pulmonary.
It always goes into the lung, but sometimes the lung walls it off, but it spreads to other parts of the body, and we haven't got a clue why.
Okay.
And most people, when it grows in the other organs like the kidney, you can also find it in the lung. It is relatively unusual to find it only extra. But we had a medical student a year ago at the medical school who had Pott's disease. He had TB of the bone and no evidence in any other organ. And, you know, had bone problems, and I think they must have been flabbergasted, you know, to find a disease. There's a mummy, a 6,000-year-old mummy in Egypt that had Pott's disease. So we know TB's been around a while.
As it's an aerobic bacteria, how long does it take to die without oxygen? If you deprive it of oxygen?
Sure. So I could also show you a marvelous slide. A guy named Cohen in the 1930s did an agar bottle and transferred TB to that bottle, sealed it, okay, and it sat for 35 years. 35 years. The agar was wrinkled and dry. There was no air, no oxygen, nothing. And somebody found it left in the incubator that had inherited his laboratory and took it and plated it out, and TB grew. So that's the point of latency. So nobody knows why the bug goes latent. The hypothesis is we know when you have enough oxygen, it grows in the upper lung. And the question is, what happens when it's deprived of oxygen? And the answer is, it goes into a dormant state. And how long it can stay, a whole slew of genes, dormancy genes, go up and down. So that's a really hot area to try to figure out what goes on. And if we had a drug for latent TB, a third of the people in the world are skin test positive. If you had a drug that killed the latent form, you would take everybody with a positive skin test, treat them for a month with the drug, and you'd wipe TB off the planet, because there's no animal reservoir. Okay.